Do your access card work to get through the door? Or it is like
no, Elise was there, so she opened it for us. That's it.
It's to keep you all awake.
I'm excited.
Yeah. There's gonna be some phylogenetic stuff.
Yeah.
Maybe. Yeah.
How are you?
I'm nervous.
Yeah, so the phase, not the phase paper, the paper that I was mentioning a minute ago, they actually looked for the cross phage and tried to find its host. And they predict that it's only associated with the bacteroides.
Ah, wow. That's a cool thing.
Yeah, it's neat that you can do that, but it's actually it's different to the result that Matt had.
Oh, really?
Yes. So Matt looked at a phase data set, and he sees links between the cross phage and like 4 or 5 other bacteria.
Can you remind me what this cross phage? It's the one that Boss discovered.
Yeah,
but what is it?
Boss and Rob Edwards discovered.
But what is this? What is this?
It's the most prevalent, most abundant bacteria phage.
Oh, okay. And they just called it like that. And it's very.
I don't know why they call it cross phage. There's a story.
Yeah.
It's kind of a funny story. Rob tells the story well. He blames it all on Boss. It was his idea.
Yeah, I should remember saying that when I mentioned the phages story. Maybe I get Boss attention again. Like, hey, yes, I want to look at your data too.
Who is your master's supervisor?
The master is composed of 9 months internship and 6 months internship, and then you have some coursework, and then you have a writing assignment. And for the writing assignment, you're supposed to spend 2 months on just literature review. So you're just reviewing literature and writing assignments, and that he was the examiner for that. So it's quite different from every master degree. It's not. I didn't do a physical internship. I didn't work. I worked. It was a literature review.
It's not like a thesis project.
No, it was a literature review writing assignment. So, and back in the day, I was just looking at inflammatory bowel disease, and I was summarizing what taxonomic groups were found to increase or decrease compared to healthy people. So it was just reading and goes up, goes down, and there was a lot of overlaps in the literature, but I wasn't able to actually take a data set from somewhere and look at it myself. So
just learning to read and write.
Yeah, read and write. Yeah, but I realized the 2030 ways that you can get biases along the road. So maybe it's worth knowing everything about that.
Oh my goodness, we're still early.
Oof, no, but can you?
That's good.
Oh, maybe they don't hear the audio. Did you enable it?
Sorry, what's that thing? There's a few more people.
Can you just mute people, but and also the video? But
yeah, I should be able to.
But I'm not sure why someone told me that I cannot be heard.
Do you need to use a microphone?
The microphone? No, it's fine. We tested it, and you could hear the voice even if I'm standing further away. Works quite well. Wow. Hello.
How many days a mile out?
Oh, depending on how many people are going to show up, I guess. Wow. So.
Tony showed up briefly.
So did you turn off the video of some people and the audio as well? Are you able to hear? Because is
Test. Test. Yeah, you can hear me. So, and you can also see me. Ora vedi. Shannon. Huh? Martin. Hello. Hello.
Okay.
Wow, it's becoming real. It's becoming real.
Good morning.
Aaron, are you there?
Brad. It's for Aaron.
Yeah, we might wait just a few more minutes for
Mm.
people to trickle in because we're in an unusual room.
Okay.
But as Barbara pointed out, people are 5 minutes late to the grid room anyway.
Yes. Good point.
Coming in on Zoom.
Aaron, this is Shannon. Can you hear me?
Yes.
Who's the assessors that you're using?
We have 4 of them. Do you want me to email you a list?
Yes, please. Yeah, just so I know who to expect.
Okay, I'll do that right after we start.
Okay, great. All right. You're more or less ready to go then?
Yeah, I think so. I don't
Yeah.
see anybody else trickling in on this end. There's a few people coming in on the Zoom still, but I can admit them as they show up.
Yeah. Well, I've made you the host of the meeting.
Okay.
So, I will bow out and let things proceed as you can run it as you see fit, Aaron.
Okay.
And I was just checking in to make sure it was all going and it seemed to be working okay. So,
Yeah, I think we're all good.
okay.
Ready to go. Thanks, Shannon.
All right. Good luck, Daniela. I'm sure you won't need it. You'll be fine. All right. Bye.
Bye, Shannon.
Amazing. Yes.
You ready to go?
Yeah.
Yeah. Okay.
Do you want to talk too?
Yeah, I'll just introduce you.
Okay.
That's all. Hi, everyone on Zoom. Thank you for joining us. Hi, everyone in the house. Thank you for joining us. I am absolutely delighted to present to you Daniela Gaio, who will be telling us about the work that she's done over the past several years in pursuit of a PhD in computational metagenomics. Danny came to us after doing a master's degree in the Netherlands and undergrad. Where was your undergrad?
The Netherlands.
Also in the Netherlands.
Yeah.
And she's originally from Sardinia, and she's the first international PhD student that I've had the pleasure to work with. So it's been a real adventure for me, understanding basically all of the interesting struggles of life in Sydney as an international student. But she's had a really intense and fantastic journey through the science, and she's sort of seen every element of microbiome science, from running a trial to sample collection, processing, generating data, and then analyzing that data. And so it's a really big journey, and she's got a lot to tell us, and I'm looking forward to hearing about it. And I'm going to get out of the way now because she really does have a lot to tell us. Okay.
Thank you, Aaron. Thank you. Okay, I'm going to start. So I'm going to talk about the generation of the largest big metagenomic data set and the insights we gained into microbiome composition and function. I'm going to tell you first how I got here. Between 2013 and 2014, I was hoarding papers about microbiome and reading a lot about it. So I decided to to join Duty with Pleasure and write my assignment for my master's degree on microbiome. And that's, I looked for the perfect examiner, and Bas Util was my examiner back then. I wrote on microbiota shifts and the potential causes of onset of IBD. And by reading about microbiome, it made me realize how important the bioinformatics behind it is. So I wanted to do a PhD that would contain the bioinformatics component, the microbiome in association with disease, and probiotics, possibly. I've been looking for it closer to home in Europe, but the few projects that I found involved rats and the dissections thereof, so I wasn't too enthusiastic about that. So I went on and typed on the Google search bar, microbiome research Sydney or Brisbane or the few other larger cities. And that's when I found Aaron's group. And it was quite a coincidence because at the same time, Aaron, Tony, and Steve, they obtained a grant to run a very large pig metagenomic study. And they got the ethics approval. And they were looking for 2 PhD students. And that's how me and Tiz, we ended up here. And we are both stubborn female islanders from Italy. And she has been working on E. coli isolates, and I've been working on metagenomics. So why is a metagenomic pig study relevant at all? Because there are farm animals, right? And they go through weaning, which is the process during which they switch from a milk diet to a solid food diet. And while in nature, weaning happens at between 12 and 17 weeks, in the farming settings, that happens at 4 weeks of age. They're forced to wean at 4 weeks of age. So what's the problem with that? During that age, their immune system is not completely developed. And the fact that they are confined in small spaces and due to intensive farming, they're more subject to develop infections. And that is majorly due to Salmonella, Clostridium, and ETEC, enterotoxigenic E. coli. So when a farmer spots the infection, he takes up the piglet and he puts them on the side, and all the other piglets are prophylactically treated with an antibiotic to prevent other piglets from getting infected. And ETEC kills very, very quickly. So what is the solution to this? In my personal opinion, the solution is to lower the demand for meat and improve their lifestyle maybe, and let them win when they're supposed to win. But that's if we would have taken that solution, we would have not probably got the grant, for instance, among the other things. And what's the big problem? The big problem is that when animals are treated with an antibiotic, and the pathogen could acquire antimicrobial resistance, but not necessarily. Also, the rest of the bacteria could acquire antimicrobial resistance while the pathogen load decreases. And bacteria do what they do best. They adapt. They transfer antimicrobial resistance into the environment. They conjugate with other bacteria, and they move the antimicrobial resistance also goes into another organism via phages. So when an AMR is in a new organism, it could either get integrated into the genome, it could also get lost, and it could also be retained in a mobile form. And that is known to provide also selective advantage, contrary to what we used to believe. So to go to the human settings, so that is majorly where you would see AMR, where you would see the consequences of AMR in society. If you take a tour in the intensive care unit of any hospital, you would see how common multi-drug resistance is among people. So that is because when a patient develops an infection, it gets hospitalized and it gets treated with an antibiotic. And actually, patients with weaker immune system, they tend to make more trips to the hospital, and so it's very likely that they develop antimicrobial resistance. And when they do, and when they have an infection and there is no other antibiotic to treat them with, they die. What does this have to do with the pig? Well, in the farming setting, as I told you, there's development of AMR due to the antibiotic use. And how does AMR spread from the farming setting into the world and to the rest of the settings, into the human environment? So via the water, and water plants don't actually have filters that filter out the antimicrobial resistance. They're not that tiny. And via manure, because manure from the piglets, they're used to as fertilizer for the crops. So even if you don't eat meat, even if you eat veggies, you end up accumulating AMR in your system. And that is the predicted number of deaths per year by 2015 due to AMR, 10 million and 8.2 million for cancer. And the World Health Organization is quite alarmed about this. So the main objectives of this project were to study antimicrobial resistance in the piglets, to study the effects of intramuscular neomycin antibiotic treatment on the guts of these piglets, because it's known that oral antibiotics have an effect, a deleterious effect on the gut, but it's not really known what intramuscular treatment does. And then there is a strong interest in finding alternatives to the antibiotics, for instance, by means of probiotics. So we started the trial back in 2017. As Aaron said, I was involved from sample collection up to the data analysis, and this is how it started. We got the ethics approval, and on 2017, 31st of January, the piglets, 126 piglets arrived at the facility. And the trial lasted 5 weeks. And weekly, we collected samples, and twice weekly from all of the piglets, and twice weekly, we collected samples from a subset of the piglets. We had a lot of samples at the end. And these are treatment groups: placebo, probiotic number 1, probiotic number 2, and that is antibiotic, neomycin. And then we had antibiotic plus probiotic number 1, antibiotic plus probiotic number 2. And the piglets were, this is important for, I'll tell you later why, but they were physically separate. These cohorts were physically separate by concrete walls. So 4 different rooms, and 2 of these rooms had the probiotic on one side and the antibiotic plus the probiotic on the other side. 6 cohorts in total. So we collected the samples, we homogenized the samples, and we stored them on the same date of collection. We stored them at -80. So my thesis contains 3 chapters. We developed and we tested the performance of a wet lab method called HackFlex. And this method played a crucial role in the obtainment of our huge data set. Result chapter number 2, I'm going to show you the results from the data analysis of the metagenomic reads. Result chapter number 3, I'm going to show you the construction of the so-called metagenome assembled genomes called MAGs, and the results obtained from the analysis of those MAGs. So, what is HackFlex? HackFlex is a method, a wet lab method, that is much cheaper than the original one. It allows you to process 8 times more samples than the original one. Michael and Joyce, they discovered the secret ingredients of this method, and Keishi made further improvements, and she's currently running a sequencing facility at UTS using this method. And I've been just testing the performance of HackFlex, of course, by comparing HackFlex with the original protocol and with another partially hacked protocol. And this is the pipeline. And this is actually, thank you, Fred, for encouraging me to use R, which I never stopped using since. There is 3 library prep methods. We have HackFlex, against SummerFlex, and the partially hacked method. I cleaned the reads, I mapped them against a reference genome, and I looked at a bunch of things. I looked at coverage, at read length, at GC bias, and contamination, and a bunch of other things. And these are some of the results. So basically what we want to see here, are they different? Is HackFlex as good as the original protocol? Well, based on coverage, yes. Also based on coverage of the low covered sites. Based on fragment size, yes. GC bias, very good. Actually, it's quite good. HackFlex is quite even better in this experiment than the original protocol. And we also tested, because the GC bias is also dependent on the GC content of a specific species. I also tested Staph aureus and aeruginosa, because one has low GC content and the other one has a very high GC content. So yeah, all good. So HackFlex performs fine, and there's a huge difference in price. The original price of the original per sample, it's over $70, whereas the HackFlex costs that much per sample. And a little added to the protocol, with using half of the polymerase, we tested the performance of this, and it's even cheaper and as good as HackFlex. So let's go back to the piglets, close the brackets of HackFlex. Why are we running this project? We want to create a large metagenomic data set that describes the microbial community of these piglets. To get the AMR profile of the piglets and the mothers prior to any treatment, understanding how the AMR profile changes during and after antibiotic treatments, describe the community profile and its development from the immediate moment after weaning, discovering if intramuscular neomycin has a dysbiotic effect, because the oral antibiotic does have a strong dysbiotic effect, discovering if and how the 2 probiotic formulations shape microbial composition. I'm going to cover this one in the next chapter. I already described to you the pig trial. I'm going to show you some of the wet lab part. And then 2 and 3, don't expect them today in this presentation, because they're part of my work right after my submission. But I'm going to talk to you about covering points 4 to 6 in 2 ways. One is with the use of reads, metagenomic reads, and one is with the use of metagenome assembled genomes. This is the pipeline. So we go back to the pig trial that I've showed you before, and we had 911 samples, and that includes the pig samples, 20 negative controls, 25 positive controls, and of these 25 positive controls, there were 3 types. We had the 2 probiotic formulations that we tested the efficacy of, we took them as well as positive controls. And then we had the mock community. I made it myself in the lab, and it contains a mixture of positive and negative gram-pos and gram-neg. And thank you, Amy, Piklu, Rosie, and Joyce for providing the ingredients of the cocktail. So I did the DNA extraction. And for the DNA extraction, we used random block design to control batch effect. And we used HackFlex for as a library prep method, and we put the samples in equal amounts. They went through a couple of rounds on the MySeq to get them normalized. And that was my first experience with coding with Aaron. We made this, we programmed the robot to do it because the robot does way better than myself. And Kay also helped with the normalization methods at a later stage, just right before we shipped the sample for sequencing on a NovaSeq. And that's where the data is, the NCBI accession number. And it was big and scary, and I was really afraid of this data set. Unlike the dummy data sets out there, big real data it's different. And we didn't always agree. We didn't always agree with Aaron on the way I would name variables or the naming of my R codes. But I did the data analysis anyway. And Matt, he contributed so much at the start because he did the reads cleaning in this part. And then I took the positive control reads and I mapped them against the MetaFlang2 database to get an idea is what we have put in the positive controls coming also out in the data. And then we pushed them also through STORME RNA. I extracted the 16S genes. And then all the reads went through PhyloSift, and PhyloSift returns alpha diversity and beta diversity values, which I'm going to cover next. So, I'm going to talk about the community composition, the development of the post-weaning piglet microbiome. And it's going to be done from a phylogenetic point of view. But I'm going to give you a brief introduction to the terminology so that you don't get too lost, hopefully. So you have 3 samples, and each sample contains bugs, and different colors correspond to different bugs. And you have phylogenetic diversity as a way to describe diversity of alpha and beta diversity. Of alpha diversity, we have richness, which is described by this so-called unrooted PD. Based on richness, sample C, because it has the most color, has the highest unrooted PD. And evenness, described by balance weighted phylogenetic diversity. The highest BWPD is seen in sample A, because you see the proportions 2 and 2 and 2 and 2, better proportions than these other than B and C. And we have beta diversity, which is used to describe how similar to each other two or more samples are, or how different. And this starts from here. You have a tree of life, and each species corresponds to an edge on that tree. And you have a data frame like that, where you have as rows, you have the samples, and as columns, you have the bugs. And you fill them up with, in real life, you fill them up with reads, number of reads, and here it's with the number of cells of dots that you see. Basically, you just take this data frame and you push it through a PCA. And this is called an edge component analysis. And we'll return eigenvalues and eigenvectors. And the eigenvectors are the edges of the tree. And it will return this PCA. It will return like any PCA principal components, where PC1 it has the highest variation, PC2 it has the second highest variation, and so forth. So this is a real life example where we have the Coligar, the Discover, the 2 probiotics, and the mock community, and we have here the number of species, the main components of the positive controls, and then we have the number of contaminants. And you see that if this is the richness for each one of them, the BWPD really drops for the Coligar because it has really the worst proportions. And if you look at the beta diversity, each dot, it's a replicate. So you see the replicates, the cluster together. And you have a cluster of the mock community, the cluster of the Discover, and the cluster of the Coligar. And if a sample ends up on the right side of the plot, it means it has more of these taxa. If it's more on the left, it has more of that taxa. More on the right, more on the top, it has more of Enterococcus faecium, for instance, which is exactly what this cover has, and you get the idea. So I'm going to tell you what did we use these PD values for with the piglets. Well, we had a bunch of metadata. But we had a few variables to look at, like the treatment cohort, the breed, the age of the piglets, the mothers of these 126 piglets. There were 42 mothers, and we had the weights measurements. They were measured, they were weighted weekly. And we had the alpha diversity, which is unrooted PD and BWPD. So basically, I joined the information. Same I've done with the beta diversity, where you have the PC, principal components, and I joined the information. And I also had the 16S genes. I ran a PCA with those genes, and I joined the information. And I would test my hypothesis using several statistical tests and adjusting the p-values in several ways, basing my decision on the variable being a continuous one or a discrete one, and basing my decision also on whether the data was normally distributed or not normally distributed. So this is the unrooted PD of the piglet. It has a peak after the first week, right at the start. It goes up because the new species, that means the new species are introduced right at the start. But at the same time, the BWPD goes down. And that means that the newly introduced species are not taking up a substantial proportion of the composition. And that's why it's dropping. And the week after, it goes up again. It goes up because those newly introduced species are starting to colonize and take up a bigger proportion. So, yeah. What happened? Sorry. Yes. Okay. So the biggest thing that you saw before, you have just looked at the alpha diversity changing over time. Well, this is beta diversity changing over time. Here you have all of the samples, and they're colored by time point during the trial. And they go from red to green, blue, dark blue, and that is what it means. It actually cost me quite a lot of work to get to extract from the XML tree these things. And that's what it means. It's not just a clustering of samples, it's telling you, oh, these samples that are on the right side, they have more of that. And when they switch at day 20 to day 30, they go more on the left, they contain more lactobacilli. And weirdly enough, maybe not so weird, if you just take the 20 most abundant 16S genes and you run a PCA, you also see at day 20 and at day 30 a bunch of lactobacilli showing up. And this is completely different data. So this is beta diversity. This is from the 16S genes. It's different input and same output. So a very strong signal was given by intrinsic factors of these of the piglets. I've run the test. I've done I'm showing you the p-value correction. And okay, so this is alpha diversity, and the blue, the dark, the black shapes. They have that is the p-value, and the red is the adjusted p-value. And on the x-axis, you have the time during the trial, and on the y-axis, that is the p-value. And that's why there is a threshold at 0.05. So you see that birthday is correlating with alpha diversity at the start. And breed is correlating with the diversity up to the 4th week. And the mother effect is detectable only during the 1st week. And if you look at the beta diversity using the principal components, you see a similar trend. The birthday is having an effect up to the 2nd week, the breed up to the 5th week. So imagine you have like 4 different breeds, and you expect there to be no big difference you don't expect that kind of signal. You're testing the treatment, but that is actually things that we didn't expect to have such a big impact. They're actually having an impact. this is the trial, and these are the cohorts. And before I looked at the treatment effect, I want to tell you that because it was intramuscular neomycin and not oral antibiotic treatment, and intramuscular neomycin only poorly diffuses into the gastrointestinal tract, you might not see an antibiotic effect at all. So I've run some tests using beta diversity values, and what you're looking at here, the red, they are control piglets, and the neomycin are the blue ones. And that is on the x-axis, it's a PC4. And on the y-axis, you have the count of piglets. And you see that whereas on the PC4, the control piglets are a bit distributed everywhere, the neomycin piglets are clustering on the right side, and they're showing more molecules, for instance, on that specific date. And on that specific date, if you compare the neomycin and the neomycin plus Discover, you see, for instance, the neomycin plus Discover showing more lactobacilli. And these guys showing more molecules again. And here, again, a bunch of lactobacilli in the piglets that received the antibiotic and then the probiotic, and then a bunch of the molecules showing up again in the neomycin piglets. And these are actually statistically significant, and I'm only showing you a few of these plots, but I didn't get too many actually of significant differences after bone pheronic correction, because the treatment, I'm going to spoil it for you, but it didn't have such a huge effect as I would have expected. And then we had the weights. So I ran some tests, and I wanted to compare the weights. Is the probiotic making the piglet weight gain more or less weight? Actually, we found this. We found the Cologuard had an effect on weight gain. So that and that. Also, the Cologuard given after the neomycin had a lower weight gain than the neomycin-only piglets. And then this is from the 16S reads, and I used weight as a continuous variable, and I used Spearman, and I found a bunch of taxa correlating with weight. And for visual purposes, I'm just showing you the piglets as groups of skinny, normal to skinny, normal to fat, and fat piglets. And what this is telling you is, for instance, that at time point 6, skinny piglets have less of the lactobacilli than fat piglets. And the streptococcus is higher in the skinny piglets than in the fat piglets. That's what it's saying. But am I actually sure about this? Recently, we have been talking about all kind of problems that can arise when you use correlation like Spearman with compositional data. And Tom, I'm very curious to see what you think about this. I'm not too sure about this stuff, but that's what we found running this test. Okay, this is, to me, the most interesting part. So we generated over 50,000 pig metagenome assembled genomes, and that made us gain some insights into the composition and function. So we're going to jump into the bins, but not before I show you what a bin is. And I wish someone would have told me right at the start, when you see the word bin or mag or metagenome assembled genome, people actually mean the same thing. And that, it would have spared me some time understanding. So imagine these samples come from the same piglet, time point 0, time point 2, time point 4. And you see that these different colors of fragments, they correspond to read, and different color mean coming from different species. And you run a co-assembly, and you're not afraid to put everything together because these reads are barcoded. So you're able to say, oh, this read here comes from time point 0, and this read here comes from time point 2. So you use this information to make this table. For each config here, you fill out with the read count. And if you look at the orange one, 167, 167. Look at the blue one, 744, 844. They are following, this is called differential coverage. So you use this kind of information to bin things together. Not you, but the binner, MetaBet 2, they do that. So they know based on the same frequency of things going up and down together, you know that it's the same organism. And this is from a real life example. This is a cartoon, but this is real. So in bin 100, all these contigs ended up together inside bin 100 because they show the same trend, basically, yeah. So I'm a very stubborn person. I was still convinced that there would be some big treatment effect to discover. And because I haven't really seen a huge effect using the phylogenetic diversity, looking at the reeds, maybe now that I have the bins, I will find something crazy. So, but you have to take into account that time having such a huge effect on our data set is practically ruins everything, because when you have a strong signal, the milder signals that are masked. And there are big intrinsic differences at time point 0 as well. So intrinsic differences among the piglets. So that is additionally adding more noise. Yeah, real life. And then there's a tons of places where you get bias from the GC genome size, bacteria being gram-positive or negative, and the impact that has on the DNA extraction, and the library size, that is a big thing. So all possible sources of bias, and the certain sample, and all these things affect the sample normalization methods. And people in the field haven't really actually come to a point where they haven't decided what is the best, absolute best method to normalize samples. And in this presentation, I'm going to show you, I've tried at least two different methods, 2 of the 3 existing methods. So you have to also remember that the microbial community acts together. You cannot really look at one thing and separately from the other. And one question that I wanted to answer was, am I actually seeing the probiotic species that are inside the formula, the probiotic formula, am I actually see them back outside of the piglets? And the answer is no. We do see them in the positive controls, but we don't see them, with a very few exceptions, we don't see them back outside of the piglets. Maybe they are in such a low count that they don't make it through. They're not detected because they're not in a high count. So we're gonna jump into the bins. This is the pipeline, and I have to thank Matt because he took care of this while I was a bit frozen, scared of the data set. He did the cleaning, he assembled them, he did the binning. And I took the bins and I have taxonomically assigned the bins using Kraken against the GTDB database, which I'm very happy with because it's not just based on taxonomy, it's based on tree placement of different bugs and average nucleotide identities. So it's probably the best database at the moment out there. And Matt, he ran the check-em analysis, quality analysis, and I pushed them through DREP to dereplicate based on average nucleotide identity. So if 2 bins are 99% identical, they are put in the same group and they're given the same code. So yeah. And then I've run the composition analysis using phyloseq and the differential abundance analysis with CMCAT, which is quite a recent tool. And I also took the proteins, the bins, and I predicted the proteins of these bins with Prodigo. And then I pushed them through for functional analysis with dbCAN against the carbohydrate enzyme, active enzymes database. And this means that the more time points you have per subject, the more bins you're going to get. And at about 8 time points per subject, it kind of starts flattening. So you could get probably 20 time points, and maybe you're not going to get so many more bins per subject anymore. And this is some of the quality. I just want to show you that out of those over 50,000 bins, we got 12,400 nearly complete bins. And that means that they are higher than 90% in completeness and lower than 5% contamination. So using the 3 different methods, so the MAG designation was different in this in here, and also the number of bins. And I took these mags and I filtered them based on these methods, and I ran it through phyloseq, and basically you just see the temporal shift again. Like this temporal shift you just see it when you look at the bin data as well, not just when you look at the read data. Yeah. And here I've used the median sequencing depth as sample normalization method, whereas here I've used the proportion as a sample normalization method. And you can see a very nice clustering in PC3, PC4, like samples from different time points really separating with 1 week from each other. And yeah, look at this one in PC2. It's like the piglets really go through a developmental stage. And and you see the clustering really neatly. And this is my favorite picture. And you're not meant to, you don't have to read the species here. But I just want you to look at the trend. So species that are up at the start and they go down after week 2, things that are practically very low at the start and they start to go up at time point 2. And this one, for instance, goes very high in abundance and goes down again. And things that go up only time point 2. And then things that go, from time point 4. And things that are very high in abundance at the start and they go lower. And you have to realize about this, that inside each of these facets, you have all of the piglets from all of the cohorts. These piglets are in different physically separate rooms. So don't think of like there's no cross-contamination. Oh, they have the same bugs because they're all together. No, this kind of regulation happens for animals that are in separate rooms. And it's like all of the piglets know that Lactobacillus amylovirus has to go up from time point 2, very high at time point 4, and then lower. And all of them, just all of them, because you have all of the data points from all of the piglets. All of them. So what is it? Oh, here you have two species of the same genera that end up close to each other, but the same genera, different species, has a different trend, and that's why it's on the bottom. And you have, for instance, this one, it's Methanobrevibacter, one sort of species, and here you have another species of Methanobrevibacter, 2 different places in the heat map showing a completely different trend. What is it? Why is this happening? It could be due to the diet. They're shifting they're weaning from this point. It could be the immune system. The immune system is known to regulate the microbiota. But my question is it actually known that the kind of regulation goes down to the species level? Down to the species level. So does that mean that if I have here 150 species, the immune system has antigens against 150 species? I don't know. I don't think people know, but I would be curious. And thank you, Simone, for talking with me about this in the last 2 months. And this is also the same time point 0 and time point 2, and you see stuff, but it has the statistical here, whereas this one has it's like visual, and here you have the significance of each change and the fold change, positive fold change, negative fold change, and it's done with CMCAT to look for associations. And the most things that change the most, they change the most between time point 0 and time point 2. But you find a lot of things changing also during the other intervals. And I was like, maybe it's a good thing that we have piglets of different ages, because it messes up a bit with our treatment. Because we're looking for treatments and we have different ages, it messes up with that signal. But maybe it's also a good thing, because we see the temporal shift. What if I take piglets that belong to the same breed, just to separate the factors? Piglets that are within the same breed, I look at those samples at a certain time point, and I pick only piglets that are 3 days apart from each other. That one is born on the 8th of January and the other is born on the 11th of January. And they significantly separate in PC1 and PC2 at time point 0. And here at time point 2, they also separate in PC1, and that's not significant. And if you take the same piglets, the same selection of piglets, and you look at you run the serum cat, you also see the tiny differences. And actually the identity of those things that go either up or down, they match with what we find here. Almost all of these ones except two. One was following the opposite trend and one was just not showing up, looking at all of the piglets. And also breed, which I showed you before, with alpha and beta diversity, the breed was a big factor, but also when you look at the beams, you see the separation here in PC2 and here in PC1. And then I wanted to look at function because inside myself, I was thinking maybe now if I look at the function, I could still find some treatment effect. And I found the time factor again, but it's quite nice because we obtained 300 protein matching against the carbohydrate active enzyme database. And that is the trend that they follow with time. And as the last box plot on the right, it's the only time point that we have of the mothers. And sometimes, like this one, CE14, it goes down and then up, and then the mothers actually have a higher abundance of that. And here you also have the sample size of both the piglets and the mothers. And for each of these piglets, because I had the data, I could also associate the protein with the species. I was asking myself, is this CE14 made just by one or a few species, or is it made by a lot of different species? And I did it like this. So these are for each one. Look at CE5. What this means is that CE5, the 76% of the time that you find CE5, it's matching against the Bifidobacterium pseudolongum genome. And 20% it's found in Bifidobacterium carinum. 3% in Bifidobacterium pseudolongum A. So, and some of them are like the blue ones or the low count. They're shared by a lot of different species, and some of them are very specific. And this kind of data is not to be found out there, like the association of these carbohydrate enzyme with the species. And thank you, Simone, for chatting with me about this recently. And this is the last plot, so hold on a second. So here you have popularity of 300 proteins. How popular is a specific protein in the piglets and the mothers? Very popular, not so popular. And here you have, for instance, if CE5, it's found here at 76, right? So CE5 at 76% of the time is made by one species. So things that are on the left of the plot, on the left side of the plot, they are made by inside the genome of many different species. And things that are on the right side, they're only to be found in a few species. Yeah. And I'm not going to cover that because I don't want to. Okay, I'm going to summarize the findings and outcomes of this project. We created a library prep method that facilitates the processing of large number of samples at reduced cost. The fact that small differences in age in early stages of life have such strong correlations with the composition could have strong implications on all microbiome large sample size studies where equally aged animals cannot be obtained. We confirm the importance of breed, maternal effect, and age in the contribution to inter-individual variability of gut microbial composition. We report mild effects of intramuscular neomycin, mainly on molecules abundance and alpha diversity, that last up to 2 weeks after termination of the treatment. The probiotic discover piglets, the microbiome correlated with the transient establishment of lactobacilli. Coligar-treated piglets displayed a clustering beta diversity and a transient lower weight gain compared to control. We found the abundance of a number of taxa to correlate with the weight of the host. And we were able to describe the compositional development down at the species level of host-winning piglets. We were able to associate over 300 carbohydrate active enzymes to specific species, and this could be of potential values to those who work in this field. We generated over 51,000 metagenomes, 12,000 of which were nearly complete. And we created the largest pig gut metagenomic data set to date, a treasure to be further examined. what I'm going to do next. Now that I'm not afraid anymore of the data, I'm going to keep swimming in this data set. And there's a few things that I want to look at first, like short chain fatty acid genes, correlation genes, and I want to map my beans against other cool databases. And then, this is coming really up next. We have a collaboration ongoing with FACE in the United States, and we're going to look at the AMR story. And we are also going to be able to look at virulence genes and phage genes. And, without support, I would have never had these results. And I want to thank the piglets. This is awesome. And then I want to thank Aaron for supporting me through this journey, Tony, Steve, the grant, ARC and Altgym, and Graham, the veterinarian involved in the project, Shima, Maggie, Liz, and Mike Lake, and Matt, huge contribution, Kay, Lee, our lab, Joyce, Michael, Kathy, Fred, Matthew, Nele, and Barbara, people at UTS, Vincent, my industry mentor, and then my family and friends and flatmates and people, beautiful people that I've met during my stay in Australia, people in the Netherlands that I really care about, and the people that I've met during the conference last year in Basel, and the people who contributed to my formation as a scientist in the past, Han Houston, Vika, Ken, Kentaro Noma, and Basti Tio. And I would like to take any questions now.
Amazing.
Thank you, guys.
Very flattering acknowledgement slide. We've got a few questions from the Zoom group, so I'm going to take one or two of those and then come to the people here. So the first question to come in is from Bas.
Oh.
He asks, you mentioned that the maternal effect could only be detected in the first week.
Yeah.
Can you please show again how we're supposed to see that? And give your interpretation of that in terms of the weaning, which is several weeks later.
Yes. The weaning happened actually at time point 0, because on the moment the piglets were moved to the pig trial site, that's the moment when they started to eat solid food. So they were taken apart from the mother at that point. And on that slide, maybe it wasn't too clear, but on the x-axis, you see time. So time point 0, it's when they're actually weaning, the moment 2 days after the wean, actually. That's the time point. And you see it here. Yeah, so that's time point, and that's the p-value. So am I covering, because probably it wasn't clear that that's time. And so basically on alpha diversity, you see that it's significant below the significance threshold. Or above the significance threshold. Okay, you get what I mean. It's below 0.05 here.
Yeah, thanks.
And you might be confused because we have two mothers. We have the sow and the nurse, because these piglets actually had, almost all of them had 2 mothers each. One was the mother they were born from, and the other was the nursing. And almost all of them had 2 mothers. So you see the effect here on alpha diversity. And actually in beta diversity, if you look at the adjusted p-value, it doesn't during the first week. You see this red here. It doesn't reach the significance here. But you see it in alpha diversity. And in beta diversity, you also see it later on, and that is time the next week. Is it?
So Bas has just acknowledged that it's now clear that time point 0 is when they weaned, that they were about 3 weeks old at that stage.
Yeah, between 3 and 4. Not even all of them were 4 weeks old. Yeah, because there was a 6 days difference among each other, maximum.
We'll take one more question from here, and then we'll go to the audience.
Yes.
Kath Burke asks, can you speculate about why the Cologuard treatment was associated with low weight gain? Has that been observed before?
Unfortunately not. We just got this. The Coligar was made by the DPI people, and it was shown to have effects on the intestinal, upper intestinal, the small intestine. That's what Tony said, and that's why it was it was promising in terms of ETEC, protecting the piglets against ETEC infection. But we didn't have an infection study. We didn't test an infection, so we don't know about that. And why lower weight gain? I don't know. And it's funny that you see it both when you compare Coligar against control, as well as when you compare neomycin plus Coligar against neomycin. So we have like two controls, two groups showing the same. But I'm not sure as to why. I really don't know. It's two lactobacilli strains in probably in a 9-to-1 ratios based on our reads. To Salivaris and Plantarum. Yeah.
We have an audience question.
Yeah.
What I'm doing is very short.
Thank you.
With regards to the high C data, and
Yeah.
considering the fact that the intramuscular neomycin isn't very
Yes.
high penetration.
Yeah, the penetration is not so high.
To see the same time
That's strong.
time point effect across the high C data instead of a drug effect.
I'm hoping I could look at.
Can you repeat
sorry.
the question for
Yes.
people on Zoom?
So you're asking if the intramuscular. Yeah, if you're gonna see the intramuscular neomycin effect on the high C data, given that it's not so strong in this data, but we see the trend. I don't know. Maybe it's gonna be more a stronger signal than what we see here. We did see this molecule thing, which I have no idea what it means actually. Why these molecules are associating with the neomycin group, and what we're gonna see in the high C. I peeked into the data. We have this collaboration going on with phase, and maybe lots of plasmids. I don't know. I have just seen some figures. I haven't haven't gone into that yet because I've been busy with this. But that's next thing. So we're gonna keep you updated on that. Yeah.
Got another question from the Zoom group from Alan Wong.
Alan.
Is wondering
Yeah.
whether you've recovered any, I'm going to pronounce this wrong, Pitesi bacteria,
Well,
Depan or Asgard archaea
oh wow.
in the mags?
In the mags, I haven't looked. I've majorly looked and seen names of things that would happen that would be abundant in high counts, because things that are abundant in low counts, I'm not sure you can extract any interpretation or conclusion from that. But I haven't looked, and if you want me to, I'll just go and look at these names specifically. But no, probably, maybe. Why do you expect them to be in pig gut? I'm not sure. Well, we can talk about this later. Yeah.
Steve, question.
Yeah.
So, Sandy, just for the design of the trial, was the intramuscular drugs given to mimic what would happen normally in the field for controlling gastrointestinal outbreak disease?
Yeah.
So is it normally given intramuscularly, or is it also given by the feed or by the water?
No, in Australia it's not it's not given in the feed. Isn't it not allowed anymore? Illegal to give antibiotics in the feed? It's
therapeutically you can use it.
Therapeutically.
But you can't use it.
Wow.
You can't use it.
As normal feed antibiotic in the no. And here I can tell you what we have done here, and it was a 5 days intramuscular, 5 days treatment once a day.
Your data would suggest that it's useless giving antibiotics.
It does have some effect, but these piglets were healthy. Probably the intramuscular neomycin is a very effective if there is an infection going on. But here these are healthy. And we wanted to look, just in this part, we wanted to look at the microbiota changing. So it may not have a big effect. We have seen some, possibly, but it was not strong. It was just, I was really hoping there would be something.
Yeah, I would have thought there would be something more as well.
Yeah, something more. I would have expected that.
Your mag list.
Yeah, and the mag.
If I can.
Yes, you can look at the mags, and we're gonna be looking at the high C stuff. So there's gonna be more about the AMR story soon. Yeah.
I've got a follow-up question on that.
Yeah.
If intramuscular neomycin is an effective treatment for post-weaning diarrhea,
Ineffective.
is effective.
If it's effective.
If it's
Yeah.
effective,
Yeah.
do your findings then suggest that the use of intramuscular neomycin would be good antimicrobial stewardship practice because the treatment has very little effect on the gut microbiome?
Yeah, well, if it has not so much effects on the, and we can see it from the IC stuff, then we can probably conclude that it's safe to use.
So,
Yeah.
So one thing that's really good. That's a good statement to make. But at the same time, there's a lot of metals used in feed.
Metals
So
used in
maybe
feed.
The
Yeah.
metal resistance genes need to be looked
Yeah.
at in your mag composition.
Yeah, we can
Because
look
they
at the.
stabilize the gut microbiome,
Mm.
whereas they may not be present in a organism that comes in as a pathogen. And so there might be a dynamic interplay there between the antibiotic and metals in the feed.
There might be, but in the feed, like there is metal. There's always metals in the feed. Yeah. Yeah, and they go together, and the metals in the feed they help the antimicrobial resistance to stay. Yeah.
That's really something to have a look at.
Yeah, yeah, we can look at that. Yeah, we have the bins, we have the IC data, so you're gonna be happy about that very soon. Yeah. Questions?
Any other questions from the audience?
Liz.
Liz got one. Last question from
Yes, this is a very simple question. Why did you choose those particular probiotics?
Oh, I didn't pick them. I didn't choose those probiotics. Actually, one that we wanted to look at is Cologuard, which is the one that was very promising from the DPI testing, but they have done the infection test trial. So they saw that it was protecting piglets from getting infected. But this is not an infection experiment. It was just more to look at how is the microbiota changing with that. And the second probiotic, the Discover, actually was a replacement to the probiotic that we were supposed to use and test, because of that probiotic, we couldn't get enough of that probiotic for the day of the trial. So it was replaced kind of like last moment. And the Discover is actually commercially available. So people use it all the time. That's why the TM. Yeah, but we were not even supposed to test that one, but just the probiotic to me. Yeah.
Excellent.
And what about the people at the Zoom? Did they?
I think that we've been through the questions.
Yes?
There might be
Okay.
one more from Boss that
Yeah.
I can't tell if it's been answered or not, but you can follow up with it.
Okay, I can follow up with that. Yes. Yeah.
Excellent. All right.
Thank
you very
you.
much, Sandy. And thank
Thank
you.
you. Thank you, guys. Thank you so much for showing up. I cannot hear what you say, but yes. Talk to you soon. Just email me some questions if you have any. Okay? Yeah? And see you soon, Vincent. Ciao, Martin. Good night. Ciao.